My name is Jiaxian. I'll introduce the paper submitted to iMeta. The title is N6-Methyladenosine in Inflammatory Bowel Disease, an Emerging Regulator of Pathogenesis and a Novel Therapeutic Frontier. This is the graphical abstract of this paper, which introduces the two key terms, N6-Methyladenosine, hereafter abbreviated as M6A, and inflammatory bowel disease. M6A is a dynamic, reversible post-transcriptional modification adding a methyl group to adenine 6th nitrogen, found on mRNA, long non-coding RNA, circRNA, and microRNA, typically within the Rach motif, 1/85 nt. As the most abundant internal modification in mammalian mRNA, it is highly conserved and a potential therapeutic target. Its regulatory system comprises writers, erasers, and readers, which control pre-RNA splicing, nuclear export, transcription, and degradation. Inflammatory bowel disease, IBD, is a chronic immune-mediated gastrointestinal disorder, comprising ulcerative colitis and Crohn's disease. This review, structured around the intestinal mechanical, chemical, immunological, and biological barriers, discusses M6A modification's regulatory mechanisms IBD pathogenesis. The overarching framework of this paper is built upon three key highlights. This review highlights three aspects. 1. Sections 2.1 to 2.4 detail M6A regulation of IBD via the four intestinal barriers, mechanical, chemical, immunological, biological, in separate subsections. 2. Section 2.5 covers the dynamic M6A landscape across patients, pediatric versus adult, disease phases, acute versus chronic, and subtypes, UC versus CD. 3. Section 3 explores M6A's clinical applications in diagnosis, therapy, and prognosis. For each of the four intestinal barriers, at least one corresponding table is provided. These tables are structured according to the M6A machinery and summarize the known mechanisms by which M6A modulates IBD pathogenesis with respect to that barrier. In the final two columns of each table, subtype-specific findings, i.e. those pertaining exclusively to CD or UC, are clearly marked, and the level of evidence for each reported mechanism is graded on a 6-tier scale. In addition, each intestinal barrier is illustrated by a dedicated figure, which depicts the principal structural or functional components of that barrier and outlines the key M6A-mediated regulatory pathways in a concise manner. Among the four intestinal barriers, the immunological barrier occupies the largest portion of this review. The immune cells are discussed in two main categories, innate immunity and adaptive immunity, with the former subdivided into myeloid and lymphoid lineages. Of all immune cell types, dendritic cells, mononucleophages, and T cells have been the most extensively studied. However, the impact of M6A modification on the function of other immune cells in the context of IBD remains poorly understood and warrants further investigation. Each of the three tables corresponds to one of the three immune cell types that have been extensively studied. Moreover, the accompanying figure primarily depicts the regulatory mechanisms by which M6A modulates these three cell populations. Section 2.4 focuses on the interplay between M6A modification and the intestinal biological barrier in the context of IBD, with an emphasis on their bidirectional regulatory effects. The detailed mechanisms underlying this interaction are summarized in the corresponding table. Figure 3 also provides a concise schematic depiction of the related mechanisms. Section 2.5 aligns with the third highlight and focuses on the dynamic landscape of M6A modification driven by IBD heterogeneity, with particular emphasis on the changes in M6A machinery observed in pediatric IBD as well as in acute and chronic phases of the disease. The final section, Section 3, of this review summarizes the potential clinical applications of M6A modification in IBD. Within this section, subsection 3.2 offers the most comprehensive discussion on the therapeutic applications of M6A, which is organized into four parts: compounds targeting the M6A machinery, therapeutic efficacy, adverse effects, and drug resistance. This review summarizes M6A regulation of the intestinal barrier in IBD and its clinical applications, advancing epigenetic insight into IBD pathogenesis. Future work should clarify core M6A mechanisms, integrating M6A-based strategies into diagnosis, therapy, and prognosis expected to benefit clinicians and IBD patients.